Hi, this is Oni.
I'm Brent, and we are your TAs for Professor Ashmore's Intro to Biology class.
And we'll be talking about photosystem 2.
So photosystem 2 takes place in the thylakoid lumen of the chloroplast. Here is the thylakoid lumen, and on the outside is the chloroplast stroma. So here's an enlarged picture of the thylakoid lumen and the chloroplast stroma. Within the thylakoid lumen, 2 water molecules are broken up into 4 protons, an oxygen molecule, and 4 electrons, which are transported to the reaction center. When the antenna complex is excited by photons from light, the reaction center becomes excited and the electron is transferred to pheophytin, which then transfers the electron to PQ and also grabs a proton molecule from the chloroplast stroma.
So after the PQ accepts the proton from the chloroplast stroma, it transfers it to the cytochrome complex, and the electrons are transported together with the PQ. So as it moves down here, the PQ is oxidized, releasing the electron, as you see here, and a proton molecule. So it keeps doing this until the lumen has an electrochemical gradient, making the inside more positive with a low pH than the outside of the stroma. So because of this electrochemical gradient, the proton pump, which is also known as the ATP synthase, allows hydrogen molecules to pass through. And as hydrogen molecules are passing through, ADP is converted to ATP by the addition of a phosphate molecule. The electron that is oxidized from the PQ gets into the PC, which we don't really have to discuss about right now.
So the electrochemical gradient is caused by differences in concentration and charge of the membrane outside versus the inside. In this picture, there's a low concentration of protons and a higher concentration on the inside, while it is more negative on the outside versus more positive on the inside, which is why it's called an electrochemical gradient, because it depends on both differences in concentration and charge. Traditionally, electrochemical gradients are created by active transport of protons moving into the membrane, and passive transport is with the gradient. So, as you've learned already, active transport is against the gradient, and then passive transport is with the gradient. The mechanism of active transport creates the electrochemical gradient, while the passive transport tries to make the concentration and the charge equal.
Okay, so the only difference here is that there is no channel transporting H plus to form the electrochemical gradient. The PQ gets electrons from the pheophytin and also grabs a proton with it, and it transfers this electron to the cytochrome complex and drops the H plus into the thylakoid lumen. For the passive transport, it also goes as normal. The ATP synthase transports H plus from the lumen out into the stroma, and as it transports the H plus out, the energy it creates is formed into ATP.
This is Peter Mitchell's experiment on the theory of chemiosmosis. This is important in showing that ATP synthase was directly responsible for the creation of ATP. In this picture, Peter Mitchell created an artificial membrane in which ATP synthase, which is found in mitochondria, was artificially added to the membrane. Bacteriorhodopsin is also added to the membrane. Bacteriorhodopsin acts as a light-activated proton pump, in which, when exposed to light, will pump protons from inside of the cell to out. When there is no light active, the proton pump will not work. So when Peter Mitchell exposed the artificial membrane to light, the bacteriorhodopsin pushed protons from inside of the vesicle to outside of the vesicle. This generated an electrochemical gradient, so when Peter Mitchell did not expose the membrane to light, the protons moved down its gradient and through the ATP synthase. Thus, in this picture you can see the H plus molecules inside the vesicle while also producing ATP molecules. In conclusion, this proved that the electron transport chain and ATP synthase were indirect of each other, and that the creation of ATP was due solely to ATP synthase and the proton gradient.